Today's lecture is going to be just going through a bunch of examples of actual transporters now that we have seen sort of generically how transporters work. So just a reminder, we have two real types here of things that can transport. We have passive transporters and we have active transporters. Passive transporters do not require energy and they just allow molecules to move in a direction that has a negative delta G, and we have active transporters that do require energy, usually in the form of ATP cleavage, and move molecules against their gradients in a way that has a positive delta G. So there are also two types of transport if we want to think about how it's exactly happening in the membrane. So the two types here are single gate and alternating gates. And so what this is a single gate one has a single thing that's going to either to open and then just things can flow through in either direction. And a pump, which is actually really what is generally called a transporter, has alternating gates. So you have one that opens, molecules can go in or out, and then the other side opens, molecules can go in or out. So because this guy just literally opens up a passage, these all have to be passive. Okay, and the ones we're going to talk about here, most single gate things are going to simply let ions through, so we're going to call them ion channels. And then pretty much all the other transporters we will see are going to be pumps. I want to be clear here, some of these are also going to move ions, so not everything where the molecule moving is an ion channel, to be clear. All right, so we are going to first talk about ion channels first. So again, they are all passive. Ion channels are all passive because they are single gate transporters. So something causes this gate to open, and then essentially this is like making a pore, so molecules are going to flow either direction. All right, so because there's nothing causing them to flow in one direction or the other, it's passive. They're going to flow in whichever direction has a negative delta G. Ion channels are actually really important. There are a lot of really important ion channels in the human body. They're a huge part of neuron signaling, so your nerves in your brain, and we'll talk about those further in chapter 12. But also there are a lot of things that we think of as toxins that are the way they're going to work, are going to attack or either bind the ion channels, something like that. So they can cause paralysis or even death if they block the signals. So the most famous one is this toxin from the pufferfish. It's also found in there are some other ones found in some poisonous snakes, but the pufferfish is the most famous one that you may have heard of. People sometimes get poisoned by this because eating this fish as sushi is a sort of dangerous delicacy, and so they eat it, but if it's not prepared properly and you have this toxin in there, it can kill you. And the way it kills you is it binds to an ion channel and prevents it from working properly. So we're going to talk about an example here of a voltage-gated potassium channel. So voltage-gated is telling us what causes the channel to open. Channels are going to fall into one of two categories, either ligand-gated, which means a molecule binds and that causes it to open, or voltage-gated, which means that when the membrane potential across the membrane changes, it opens. So that can be in either direction. It can be that when the membrane potential gets lower, it opens, or when the membrane potential gets higher, it opens. But for our purposes, we're just going to talk about it changes and that opens it. So really all this does is we have a bunch of alpha helices in here, and when it's open, there's enough space for a potassium ion. There's sort of like a hole in here. And when it's closed, they sort of shift so that there's literally just not space for the potassium to pass through. And there are a couple other important things to talk about here. There's this S4 helix, and that is going to be what senses the voltage difference. So you see all these pluses. It has a whole bunch of arginine residues on it. And if the membrane potential changes, some of these are going to change their protonation state, and that's going to cause a shift in conformation that causes us to shift between open and closed. An interesting and somewhat surprising thing to some people is that the potassium channel does not transport sodium. And so in case you've kind of forgotten what the periodic table looks like, potassium is actually a little bigger than sodium. So sodium is up here, potassium is right underneath it on the periodic table. So you could maybe see how a sodium channel wouldn't transport potassium because maybe it's just the right size, but the potassium channel doesn't transport sodium either, which is the same charge and just a little bit smaller. So that's potentially a little bit surprising. So the reason for that is that this is how the channel works. So there's a little, this is the inside, and the potassium is generally going to be flowing outside because potassium is usually much higher inside cells than outside cells. And so being passive, we will see that it flows out when we open the channel. So there's sort of this large vestibule, and the potassium with its water molecules solvated can go in here. And it has to let go of the water to go through, but it'll do that because there are some negatively charged amino acids here that can interact with it. And then they sort of passes on through, and once it gets on the other side, it picks up more water, and now it's happy. All right, so yeah, this dipole here of this helix, where there's more negative charge here, it essentially attracts up at the potassium to come in there. But again, it would probably attract the sodium too, right? So why doesn't the sodium come through? And the answer is because these negative charges are going to come as close as they can to the positive ion in here. And so if it's potassium, that's exactly the right size. If it's just the slightly smaller sodium, these shift in a little bit to reach the sodium, and that causes the whole pore to collapse, and then nothing can move through until the sodium pops back out. So, which is quite a clever situation. It also doesn't hurt that potassium is higher on the inside, sodium is higher on the outside, and so the potassium is trying to go out, but the sodium would be trying to come in. But still, it's that collapsing. So if one does make it in, if a sodium is like, I'm going to go through this channel, it sort of collapses and says, no, you're not going to. It sort of holds it there until it pops out, and then it can reform, and then the potassium can come on through. All right, so then transporters, which are going to be those pumps with the two alternating gates, these can be passive or active. So if you know something only has one gate, if it is an ion channel, you know that it is passive. If you know it's a pump, you do not know whether it is passive or active. It may be using ATP, or it may just be allowing something to come in and then open out the other side, but no ATP involved. So the first one we're going to talk about is GLUT1. This is a glucose transporter. It's called GLUT1 because there are a number of slightly different glucose transporters. They are all passive. GLUT1 specifically is found in erythrocytes, red blood cells. And what it does is brings glucose into the red blood cells. So it's going to do so when it is higher outside in the blood than it is inside the blood cell. And glucose is the main fuel source for red blood cells, and so as soon as it gets in, it starts getting broken down, so that keeps the concentration low and keeps the passive transport moving in. So this GLUT1 it forms a pore. It has these 12 helices here, and it forms a pore that is polar. And so some of the helices, 4 of the helices, are going to form this pore. So they have these sort of amphipathic helices, like we've talked about before, where this part is nonpolar and this part is polar, so that the glucose can pass through it. And this is just showing you how that could, how this happens. This one, it's just one of these helices where here we have the polar side and then the nonpolar side. So this is the proposed mechanism of GLUT1, and really all this is saying is whether or not glucose is bound affects the conformation. So there are going to be 2 different conformations, T1, where it is open to the outside of the cell, and T2, where it is open to the inside of the cell. So when it's in T1, glucose is attracted to this. It probably right in here is going to have a lot of things that are going to hydrogen bond to that glucose. So the glucose goes in. But glucose binding, and now we've bound glucose there, glucose binding causes an equilibrium shift to the T2 state. But once it's in the T2 state, now the glucose can sense, essentially, again, it's not conscious, right? But the glucose concentration here is pretty low, so that's going to cause the glucose to come out. But once the glucose unbinds, now it's going to cause a shift in the conformation back to the T1 state, so it's ready to pick up another one. So reasonably simple, but you can consider, right, so these are the two gates. So you have a gate here and the gate here. So at no point is it just open so glucose can flow through. Although this is a passive thing, so it would have been okay if it did that, but it just does not. The chloride bicarbonate exchanger gives us an example of why we might have some that sometimes move more than one type of molecule. So this is also a passive transporter. And the goal of this transporter is to move CO2, it's to move, let's back up. So when you have respiring tissues, so you're going for a jog and your legs are using all the energy and producing CO2 that needs to get back up to your lungs so you can breathe them out. CO2 is another one of those things that can actually pass through the membrane on its own. It's small and nonpolar, so it can pass on through into the erythrocytes, at which point then carbonic anhydrase converts it into bicarbonate, the bicarbonate ion. And then the bicarbonate ion is going to pass back out of the cell and then be transported through the blood plasma, just like go on through the blood to get back to the lungs. But once you get to the lungs, you don't breathe out bicarbonate, you breathe out CO2. So the process needs to reverse. So it goes back into a cell, gets converted back into CO2, which can then just diffuse out. All right, the reason for this is just that it is easier for the bicarbonate to transport through the blood than it is for the CO2 to transport through the blood. Also, remember, this is part of the pH, it's part of the buffering system. So it's important that there's some of this dissolved in there. So the issue here is that although this is a passive movement, the bicarbonate out, there's more bicarbonate in and less out here, this is negative. And so if you're consistently moving negative things out of the cell, you're going to change the membrane potential. If you change the membrane potential, that's going to change whether or not this is passive or not. So what this exchanger does, every time it brings the bicarbonate out, it brings a chloride in. That makes the net charge difference 0, because you brought one negative thing in, one negative thing out. And then the same thing happens over here, right? If it's bringing the bicarbonate in, it moves the chloride out so that we do not have a change in the membrane potential. So that's actually one common reason why we might have co-transporters, is to keep the charges the same. All right, so now we're moving on to active transport. So everything from now on is going to be an active transporter. But there are two types, and one of them is a little trickier. So the more obvious one is primary active transport, and then that means the actual transport itself clearly cleaves ATP and uses that energy to move a substrate across the membrane against its gradient. So that's easy. That's what I've given the impression is active transport. There's another option though, and that's to just include something else in there. So this is a little more complicated. So here we actually have two transporters. One of them is obviously cleaving ATP and moving something through. And then the other one though is not cleaving ATP, but is only working because of what's happening with this guy. So here, this one on the right is allowing S1 to move through and also pulling S through along with it. It's a co-transporter. And the S2 is moving against its concentration gradient, but S1 is moving with the concentration or the membrane gradient. And so overall, the delta G of moving both things in is negative. But the only reason that's true is really because we spend all this energy moving S1 out against its own concentration gradient, or membrane, or both. So this whole system together is called secondary active transport, because without cleaving this ATP, you couldn't bring the S2 in, even though it's not the same enzyme doing it. All right, so first we're going to talk about some primary ones, and there's a category called the P-type ATPases. These are active transporters that move cations through. So again, I want to point out here, they're going to move ions, but they are not ion channels. And we can see here they have a nucleotide binding domain and a phosphorylation domain. That's where the ATP is going to bind to get cleaved. And that's going to cause a conformation change. So the phosphorylation domain has an amino acid on it that's going to get phosphorylated by the ATP. So ATP, it's not going to cleave off phosphate into free phosphate. It's going to move the phosphate onto the phosphorylation domain, but that's okay. It's still cleavage of ATP, right? So they all look pretty similar. They have a bunch of these membrane-spanning helices, and they have an actuator domain, and then we have this nucleotide binding domain and this phosphorylation domain. So this is a whole category of them. There's at least 70 important ones in the human body, and we're going to talk about a few examples here. So one of these is called the SERCA pump. SERCA stands for the sarcoplasmic endoplasmic reticulum calcium ATPase, so for each of the letters. So this is happening in the endoplasmic reticulum, and it's moving calcium. All right, so we're going to talk about how this one works. The sodium potassium ATPase is actually one of the most important, I would say. It's debatable how to classify something as important, but I think it's probably one of the most important transporters in the human body. And there's just a few others. We're not going to talk as specifically about these. But you can see just by looking at them, they all look somewhat similar, a little bit different maybe, but they all have this bundle of helices going through the membrane, even though they're all a little bit different. They all have an actuator domain shown in blue. They all have a phosphorylation domain. They all have a nucleotide binding domain. So although they are not the same and they have specificity differences, right? So this one only moves calcium. This one only moves sodium and potassium, but, and then this one moves obviously protons, and this one moves protons and potassium. So they're going to let different things, move different things through, but they all have a similar setup. So this is the SERCA pump mechanism, and I think it can be a little confusing, so let's just carefully go through it. So the first thing that happens is that 2 calcium ions are going to move in. So this is a co-transporter, but it's moving 2 of the same thing. So every cycle it's going to move 2 calcium ions. So it's got 2 gates. So this gate on this side is open, so the calcium can move in, but it's still going to keep this part closed, so it can't just go on through. Calcium moving in then actually causes ATP to bind in the nucleotide binding domain, and they're not quite showing you all of the steps here. So when ATP binds, it causes a conformational change of the nucleotide binding domain and the phosphorylation domain that causes then this to close up. So now we've got this closed up, so the calcium is sort of stuck in here. It can't go back out. That's important because it's active. If given the chance, it's going to want to go back to where it's lower concentration once we open up this side. So that's why we can't just open both sides at once. And then this guy gets cleaved into ADP, and the phosphate gets moved onto the phosphorylation domain. That causes a conformational change again of everything moves, and that opens up this channel. So the calcium is going to flow on out. And that also then, that's going to cause the active actuator domain to move, causing the ADP to get released. And then the next thing that's going to happen is this, we have to get this dephosphorylated. So that's going to get dephosphorylated here so that we can start over again. And everything just resets so that we can go back through again. So just like enzymes, we have to get back to where we started so it can keep doing it. So the sodium potassium ATPase does a similar thing where, the things are going to go in and then we're going to have shifting and ATP cleavage or whatever. But what this does is this is going to, every time it moves things through, it's actually going to move 5 ions. So it's an antiporter. It is going to move 2 potassium ions in and 3 potassium ions out. So this is against both of their concentration gradients. We can see that potassium is higher on the inside than the outside, so moving in is against the concentration gradient. Sodium is higher on the outside than the inside, so moving it out is also against its concentration gradient. The potassium movement is with the membrane gradient, but the sodium is against it. And so the overall effect of that is this movement is going to be a positive delta G. And this is actually what creates that membrane potential that I've been talking about. Because every time we do this, we move 2 positives in and 1 positive out. That is going to make the outside more positive and the inside more negative. So we need to have something to make that. This is what is going to make it. So a lot of other things are going to rely on it. This takes so much energy that when you are at rest, so if you're sitting in front of your computer right now, you're basically at rest. About 25% of our energy when resting is just being used to keep this potential up in all of our cells. 25%. Obviously, if you're doing something that's very energetically costly, you're going for a run or something like that, a smaller percentage of your energy used is for that because you're using more energy for something else, but you still use a lot of energy. So a lot of just the calories you eat every day are simply going to maintaining the membrane potential in all of your cells. Our next little category are V-type and F-type ATPases. The V here stands for vacuole. This is used to acidify vacuoles. So the vacuoles are a much lower pH than, they're just little vesicles inside the cell, and they are much lower pH than the cytosol. So in order to get that, you got to move protons in against the proton gradient there, and so that's going to take energy. We're actually not going to focus too much on the V-type ones, but the F-type ATPases, the way they work is there's two main parts to it. We have this upper part, and that's where the ATP gets cleaved, and when the ATP gets cleaved, it causes this whole thing to move around. And moving this around, it's literally linked like a sort of axle on a car wheel to this bit, which is in the membrane. And when this part moves around, this part moves around, and that pushes, there are going to be protons that are going to come through, and the physical motion of moving it is going to push the protons to the side that is higher concentration already of protons. However, the actual main reason we will eventually talk about this is that if you see actually there are 2 arrows going both directions, and that might be a little confusing. The most famous F-type ATPase is not actually an ATPase at all. It is one that works in reverse, and it actually uses protons moving through with the proton gradient to make ATP from ADP. So ATP synthase, which is responsible for making the majority of the ATP that is then used for everything else, is actually a backwards running F-type ATPase. So we'll talk more about how that works when we get to that section, which will be in the metabolism section. It'll actually be one of the very last days of class. All right, so then another category are ABC transporters, and these again are a class where we have large multi-helical structures in the membrane, and we have a couple nucleotide binding domains. So again, we can easily see that these are going to be active transporters because they have a place to bind and cleave ATP. Some of these are very specific. They're only going to maybe move one specific molecule, but some of them are actually much less specific. There's an important one in cancer that's called the multi-drug resistant transporter. And what that does is essentially it sees molecules it doesn't think should be inside the cell, and it moves them out, which may seem like a really important thing, right? Except if it's expressed in a cancer cell, and the molecules it's moving out busily are the cancer drugs that are supposed to kill the cell. So this can be upregulated in certain types of drug-resistant cancers, and it's basically working against any chemotherapeutics that are working, because it's basically, you're giving it the chemo, but if it can't stay inside the cell, it's not going to do anything, and it keeps the cancer cells alive. Another important ABC transporter is important in cystic fibrosis. So cystic fibrosis it's a disease of the lungs, and what happens here is they get this really thick mucus in their lungs, and eventually that you can't breathe, and that's obviously problematic. Breathing is important. And so the reason this happens is that this transporter is defective. And this is a chloride transporter. It's supposed to be moving chlorides across a membrane. So it's closed, and then it will bind ATP, change the conformation, allow some chloride to move chloride through, and then reclose again. However, in people with cystic fibrosis, there is just a mutation. It's actually a single point mutation. It's a deletion of a phenylalanine. It causes the protein to be closed all the time. And so then you can't move chloride transport, you can't move chloride through, and having less chloride through causes an imbalance in osmotic pressure, causes a water issue, and it causes mucus to grow, and then you get bacteria growing and things like that, and it's kind of obviously a big problem. And this all just comes from a single mutation in an ABC transporter in the lungs. So ion gradients that have been created from other active transporters, a lot of times this is going to be the sodium-potassium ATPase, but it could be other ones, can then be used in a co-transport system where we move the sodium or sometimes the protons with their concentration gradients and move something else against theirs. All right, so here we're going to this is what we want to transport. And then we're going to co-transport a solute that moves in a way that is a negative delta G for it. But this is considered secondary active transport because the only reason those have those concentrations is because of another active thing like the sodium potassium ATP transporter or potentially one of those proton pumps. So we're going to see a couple examples of this as well. So one of these is the lactose transporter. This is another example of a class, so it's part of another class of transporters called the major facilitator superfamily. So again, there are others. So the way this works is we're going to move lactose from the outside to the inside of a cell. And while that's happening, we're going to have co-transport, symport of a proton into the cell. That is going to be favorable, although the lactose is going to be unfavorable. And there's another proton pump that is then going to turn around and move the protons back out of the cell so it can keep going. So the protons are basically doing this. And this is what makes it secondary active transport. This is also actually an example where it's not just an ATP cleavage. There's a different energy source here. So I don't want you to stress about that, but just to point out, it doesn't always have to be ATP cleavage, although it usually is. And this one looks kind of a little bit like the GLUT transporter, although obviously it's going to be active, right, where we sort of have this rocking motion that's going to allow it to shift. Second example is the sodium glucose symporter. This is found in the epithelial cells in your intestines. So what happens here is, so this is the inside of your intestines, and what we need to do is we need to move the food that you ate and that's been broken down in your intestines through those epithelial cells, eventually into your bloodstream so they can be used, right? So here we have glucose, and the glucose concentration here is going to be higher than in the lumen, so that's going to need active transport. So we can't just do it on its own, so what we're going to do is we're going to pull two sodiums through. Remember, sodium is higher on the outside than the inside of cells, so that's going to be favorable. And so this only works, these co-transporters only work when you move everything at the same time. You cannot just move just the sodiums without a glucose. So essentially the glucose comes along for the ride because the sodiums really want to come in. So now the glucose is in here. Now we can have a passive transporter here, a regular glucose uniporter. This is GLUT2. It looks very similar to GLUT1. So this is passive. And that's because we have a high concentration of glucose in here, higher than in the bloodstream. So this one is active, and that one's passive. And then this is showing you here, this is how the sodium is then getting moved back out of the cell, and this is just the sodium potassium ATPase. And the last thing I want to talk about here is the fact that water actually needs a transporter. Water obviously cannot go through those membranes, and most of the things we've seen do not allow water solvated things to move through either, right? We had to let go of the water to move through. So we're also going to have a transporter for water in the situation that we need to move water through. There are plenty of reasons you might want to do that. And these are also just going to be, sort of little channels that allow water through. You almost at that point can think of water as being like a solute, even though of course it's also a solvent over here. But from the aquaporin's perspective, it doesn't really matter that it's moving solvent molecules. It's just moving molecules. And these aquaporins actually have some charged things on the edges, and that is so that only water itself will move through, and we won't get like hydronium ions, H3O+, or hydroxide ions, OH-, right? We know those are going to be around to a little extent in there, and so we're only going to move the neutral waters through. And that is the end of that lecture and the end of chapter 11.